NCE Advanced Level Chemistry Practical Series, experiment number 33. Experimental study of the characteristics of a dynamic equilibrium with the use of ferric ions and thiocyanate ion equilibrium system. The aim of this experiment is to develop a better understanding of a dynamic chemical equilibrium. Furthermore, we are going to observe how an equilibrium behaves when it is applied with certain stresses. Without further ado, let's move on to identifying the reagents we will be using for this practical. Let's take a look at the reagents we will be using for this practical. We have over here our ferric nitrate solution, which we have prepared by dissolving ferric chloride in nitric acid. When it comes to ferric salts containing Fe3+ ions, when you dissolve them in water, you normally observe a yellow-brown color solution. This is due to Fe3+ ions deprotonating the water ligands that are attracted to it and forming ferric hydroxo complexes. These are usually yellow-brown in color. So if you add an acid to this deprotonation would be reversed, and therefore you will form water ligands, or surrounding Fe3+, you will have water ligands, and this aqua complexes or hexa-aqua Fe3+ complexes will be brown in color. So this is why you observe the Fe3+ ion solution that we have here to be brown in color. And then we have our potassium thiocyanate, and we have our sodium hydroxide. We have another beaker for the control solution, and we have 4 test tubes labeled 1 to 4, so that we can very clearly identify which reagent has been added to which test tube. We will be using droppers to add reagents as well. Now, let's move on to our practical. First, let's take the beaker that is labeled control. Now, let's move on to adding ferric ion solution to our potassium thiocyanate solution. So let's first take our ferric nitrate solution that we have here, and let's add it to our beaker here that is labeled control. And then to it, we'll add a little bit of potassium thiocyanate. Now, potassium thiocyanate, when reacted with Fe3+ ions, will form a blood red color solution. Let's observe the formation of this blood red color solution. We can see here, when Fe3+ ions comes into contact with thiocyanate ions, it forms a blood red color solution. Now, let's add water until we observe an orange color in this beaker. It is still red in color. We need an orange color. As we add water, we are diluting the solution. As this solution is quite concentrated, we can take another beaker and dilute it a little further. As you can see, now we are reaching an orange color. Now we have diluted our blood red color solution to an orange color solution. When we are starting off, the Fe3 plus ions were brown in color, but as you know, as time passes by, when more and more deprotonations happen, Fe3 plus ion solution eventually end up looking yellow in color or yellow brown in color. As you can see, even our ferric nitrate solution is no longer that brown. It is changing color as more and more deprotonations take place in the aqueous medium. So the initial solution when Fe3 plus ions are present in the medium was yellow in color. And then when you are adding thiocyanate ions, what happens is due to the reaction between Fe3 plus and thiocyanate, we produce iron thiocyanate complex, thiocyanate iron 3 ion. So this complex is going to be blood red in color. The mix between this blood red color complex and the yellow color Fe3 plus complex is orange. So, or this particular beaker with the controlled solution in it, you can see the color of the solution is orange. This is due to the presence of a yellow color complex as well as a blood red color complex together. Both of those colors together appear orange. Now what we are going to do is we are going to remove 4 portions of this solution into these 4 test tubes. Like so, we'll take one test tube and pour a small amount of the solution into each of these test tubes. Now we have added our control solution into 4 separate test tubes, and in these test tubes we will not add anything to the solution in the first test tube or the test tube labeled 1. So it is easier to compare the colors when they are all in test tubes. We will not add anything to the test tube 1. So test tube 1 will act as our control solution in this test tube rack. To the second test tube, we will add a little bit of sodium hydroxide. As you can see, upon addition of sodium hydroxide to this test tube 2, we can see the color of the initial solution changed slightly to a lighter color, more towards yellow. The control is here. You can see if you're taking a look at the control test tube that we have here. The control test tube has an orange color, and after adding sodium hydroxide, the color of the solution in test tube 2 appears to be a little yellow in color. Make note of these observations, and then we move on to test tube 3. To test tube 3, we will be adding a little bit of ferric nitrate again. Observe what happens to the color of this test tube, of the solution in this test tube. As you can see, when you keep adding ferric nitrate to this control solution, you can see the color changed from a lighter orange color to a darker orange color. If you compare it against the control solution we have here, you can see very clearly it has gone from this orange color over here more towards a red color or a darker orange color. Now lastly, let's move on to our test tube 4. To that, we will add potassium thiocyanate. Let's take a few drops of potassium thiocyanate and add here. As you can see, when even a single drop of potassium thiocyanate is added, you can see the color changing more towards a darker orange color. The initial solution was a lighter orange color, but when it comes to the second test tube, or the fourth test tube rather, that we have here, is a darker orange color. Let's add another drop to see if the color changes a little bit more. As you can see, the more potassium thiocyanate we add, the closer it will get to a blood red color or a red color. It is moving away from the orange color that we have here, and it comes closer to a red color. Now students, let's take a look at what has happened here. Let's take our ferric nitrate solution here. The color of the ferric nitrate is slightly yellow in color or yellow brown in color. And when you added potassium thiocyanate to it, we turned this yellow brown solution into a blood red color. So you can see these are the two colors that you observe in the two extremes of this equilibrium. Before adding potassium thiocyanate, the solution looks more yellowish, and after sodium thiocyanate has been added, the solution appears more blood red in color. What we have obtained here is a small volume of this blood red color solution, and we have diluted it until we obtain an orange color, a color that is in between these two extremes of the equilibrium. And then to this orange color solution, we added a little bit of sodium hydroxide. And when adding sodium hydroxide, you can see the solution turns more towards a yellow color, and even a small precipitate of iron 3 hydroxide or ferric hydroxide has been formed here. This is the ferric hydroxide precipitate you see at the bottom, and the solution is more towards a yellow color. As Fe3+ ion concentration goes down, equilibrium position shifts more towards the left hand side, producing a more yellowish color in the solution. If the equilibrium position go more towards the right hand side, where iron thiocyanate complex ion is formed, the solution would look blood red or closer to blood red color, because the two extremes of this equilibrium is Fe3+ aqueous ions, and on the right hand end we have iron thiocyanate complex, iron 3 or thiocyanate iron 3 complex. So if you take the control that we have and add a little bit of ferric ions to it, then in the equilibrium the ferric ion concentration will go up. This stress that is caused to the equilibrium will be remedied by making the reaction go more towards the right hand side, producing more and more ferric thiocyanate complex. So you can see this is evident by the darker color that is present in this test tube. The control is light orange, but test tube 3 is a darker orange, more towards a red color. This is due to the formation of ferric thiocyanate complex. And in test tube 3, we increase yet another reactant concentration. The concentration that we increased in this particular experiment was thiocyanate ion concentration. So thiocyanate ion was one of the reactants we had. So when we increased the reactant concentration, we started producing more and more products to relieve the stress that is added on to the system by increasing the thiocyanate concentration. So therefore, the thiocyanate concentration goes up. To counter that, the equilibrium position will shift further to the right hand side, producing ferric thiocyanate complex, which is blood red in color. So there's a more of a production of ferric thiocyanate complex, which is going to be blood red in color. So the color over here is darker compared to our control, as you can see. This indicates the equilibrium has shifted more towards the product side. When we were adding sodium hydroxide, equilibrium has shifted more towards the reactant side, because by adding sodium hydroxide, we have decreased the concentration of Fe3+ ions in the medium. This is a summary of this experiment. Now, let's move on to our questions and important theory parts revolving around this experiment.